Hi, my name is Neeraja, and I'm a fifth-year bioengineering PhD student in the Sean Wong Lab in the Materials Science Department here at Stanford. Today, I'd like to briefly introduce my research on how to measure the influenza immune response using giant magnetoresistive, or GMR, sensors. First, I'd like to thank my PI, Dr. Sean Wong, and my collaborators in the School of Medicine, Dr. PJ Utz and Dr. Pravesh Khatri, along with my collaborator from the NIH, Dr. Luis Franco. During a pandemic, the importance of testing is crucial. When a patient comes in with flu-like symptoms, today it could be COVID, but it could also be the flu or another respiratory viral infection. Traditional testing for diagnostics usually tests for the presence of the virus itself. But what if instead we could test for the body's immune response to the virus? This genetic response would be disease-specific, and it could also tell us not only if the patient was infected, but also how well the body was actively fighting the infection. To address this new perspective, my colleague Dr. Pravesh Khatri performed a meta-analysis and found these 11 key genes that are specifically overexpressed when your body has the flu and is working actively to fight it off. In my PhD, I've developed an assay to measure the expression of these key flu genes using GMR sensors. These sensors, seen here, are on the scale of millimeters, and importantly, there are 80 different sensors on one chip, meaning that you can look for many different biomarkers in one test. These chips can be conjugated with DNA probes that are complementary to the flu genes that we are looking for. If these genes are present, they will come and bind to the probes that are on the chip, and the genes will successfully hybridize to the probe. Then we can measure a magnetic signal indicating how much of those genes were present, or the concentration of the genes. Ideally, someone with the flu will have a higher concentration of those genes present compared to someone without the flu. To actually test this assay in practice, we were lucky to get an RNA sample set from the Baylor College of Medicine with patients with flu-like symptoms coming in at day 0 of their infection. Then we were able to test these patients using our GMR sensors and measure the IMS score, or how much of these flu genes were present in each of the patients. We were able to successfully distinguish patients that had the flu from patients with other viruses, such as rhinovirus, or patients that were symptomatic but ultimately did not have a virus that was tested for in the PCR panel that created these expressions. So then we were able to develop a logistic regression model and see that we were able to accurately identify patients with the flu with an AUC of 0.92. Our next steps are to see if this gene set is indicative of effective vaccination. So with that, thank you for your time, and please let me know if there are any questions that I can answer.